Hello, everyone, and welcome to today's webinar titled mRNA Quality Assessment and USP Guidelines, hosted by Biopharma Webinars and presented today by John F. Cipollone, who's PhD, team lead, and senior principal scientist in global biologics at US Pharmacopoeia. So, John, hello there, welcome, and thank you for joining me today. It's a pleasure to have you here.
Okay. Good morning and good evening, wherever you are.
Great.
I will share my screen.
Okay, well, while you do that, I will just quickly introduce you. So John F. Cipollone, prior to joining USP, founded and led the Vaccine Structure Group at US FDA CBER. And his group focused on vaccine antigen structure, pathogen interaction with the host immune system, and development of mass spectrometry based and other analytical methods, as well as supporting informatics. So he served as a CMC reviewer for a wide range of vaccines for over 15 years. And John's role at USP includes leveraging his experience in vaccine research and review to lead efforts towards identifying and advancing the development of documentary and physical standards to support vaccine development and manufacture. His efforts also include educational outreach to foster greater understanding of the principles of vaccine development, production, and quality as their importance in human health. Okay, but now please let me hand over to John, and then later I will introduce you to Khaled as well. But for now, over to you, John.
Thank you for that marvelous introduction, Marcus, and welcome everyone. So today I'll be talking to you about messenger RNA quality assessment and the USP guidelines that we've put forward. And you'll see sort of the development of that and what it encompasses, and some of the basics about messenger RNA products and some of the market attributes. So the agenda will essentially be the introduction to the USP, what we do, what the outreach we have, and so on. I'll then talk about the messenger RNA drug marketplace, where it's gone over the last several years. Supporting vaccine quality. I'll describe how USP is doing that as part of our mission. Messenger RNA guidelines and analytics. I'll discuss messenger RNA vaccine quality attributes and analytical procedures. And then I'll summarize and provide some future directions on where USP intends to go with the messenger RNA vaccines and related products.
Okay, very quickly, John, let's go into full screen mode on your PowerPoint here.
You're not seeing the actual presentation?
It's not in reader view yet. We're seeing the whole application. If you just click on the reader view on the bottom.
Oh, I'm sorry. Let me switch. Is that working?
Not yet. Do you want me to bring it up for you? I can do it on our platform if you want.
Let me see what I can do here. Sorry.
Yeah. It's okay.
Sharing.
It's definitely sharing. It's just not full screen. Typically, the reader view on the bottom right-hand side of the PowerPoint is helpful. If you just click that.
Is that better now?
It hasn't changed. Are you using more than one screen?
Yes.
That would be why.
I'm trying to switch displays so it swaps. Maybe I'll duplicate. If it duplicates, maybe that'll work. Hold on.
Okay.
No, I can't. Oh. Sorry.
It's okay. Don't worry, John.
How about now?
Very good. Yeah, that's good.
Okay. All right. My apologies, people. So continuing. Some some information about USP's mission. So USP has been in place for over 200 years and provided public standards in medicine, dietary supplements, and food to protect patient safety and improve public health. We're an independent scientific nonprofit organization focused on building trust in the supply of safe and quality medicines and food supplements and so on. As USP continues to adapt, grow, and evolve with science and medicine, we strive for a world where everyone trusts the medicines we rely on to save lives and preserve health. USP works globally with our collaborators to help ensure vaccines are stored, transported, and administered properly. And I'll show you some slides on some of our recent work that's helped to provide some of that information to the public. USP works globally with our collaborators to help ensure testing and public standards are available to verify safety and efficacy and so on of vaccines for patient safety. So these are some recent toolkits and guidelines that we produced, and this really occurred over the pandemic period, roughly from 2021 through to present. So we've provided quality attribute toolkits, which allows for assessment of vaccine quality, vaccine handling guide, vaccine handling toolkits for primarily focused on the COVID-19 vaccines. And then we put out draft guidelines for, of course, analytical procedures for mRNA vaccine quality, and also for analytical procedures for viral vectored vaccine quality. These are now the mRNA vaccine qualities. The second edition is out. We're now working on the third. I'll discuss that a little bit. And the second edition of analytical procedures for viral vector vaccine quality is soon to come out, probably in the next, I would say, 60 days or less. So messenger RNA marketplace. There are not, but I'm not just describing just vaccines here in these successive slides. Messenger RNA-based drugs encompass a wide range of applications. So we're talking about vaccines. We're also talking about cell therapies, gene editing, therapeutic antibody production, as well as protein replacement. And those encompass a range of different areas. Vaccine development for prevention of infectious diseases, also cancer. Cell therapies, CAR T cell therapy, treatment to develop and target disease relevant to cell types. And I believe the FDA has just approved their first CAR T drug product. Therapeutic protein production, messenger RNAs can be translated into the patient's own cells and produce therapeutically active proteins. And genome editing, such as protein encoding messenger RNAs, can be used for antigen presentation, functional protein presentation, protein expression for target gene modification, and also for protein replacement for instance, enzyme deficiencies, also active area, for instance, in diabetes. So there's a wide range of applications for this technology. And this is a little more sort of focused on that and kind of spread out over a time span, and then what is actually targeted as far as diseases and syndromes. So you can see in the top left, the industries is really heavily investing in messenger RNA vaccine trials. And you can see in 2023, as compared to 2021, there's been a vast increase in capitalization of this area. And that's really moved more into sponsored by industry capital. So up basically 85%, up from just 34% in just a couple of years ago. So you can see that capitalization has really increased quite vastly in the past several years. There are, we can count over 140 clinical trials using messenger RNA vaccines currently. 70% are focused on infectious disease. That's another 12% in oncology. And you can see up on the upper right, the last 17 or 18% are kind of smattered across a wide range of applications listed as other there. And then some that target central nervous system diseases and also immunological diseases. So conventional infectious disease vaccines are available, serve as benchmarks to messenger RNA vaccines. So what do I mean by that? Well, probably this community present in the audience probably understands that when developing a vaccine, there has to be a comparator. And so primarily what's being targeted, there are already existing vaccines, so that can serve as comparators in clinical studies. Messenger RNAs have been shown potential as therapeutic for protein expression and in treatment of diseases such as cardiovascular disease, and as I've already mentioned, diabetes. And this platform is, or this vaccine class is platformable, meaning that the process is very similar across products, and essentially is adaptable to new technologies and possibly continuous manufacturing. So it's very attractive in this way. And it's kind of represented by the large influx of capital. This is another view of the landscape of messenger RNA vaccines and therapeutics. You can see on the left the status of that range of products that are in the pipeline. The probably less than half, slightly less than half, are in preclinical trials. There are a range that really there's no reported status of their step in the review process and development process. There are approximately 40 to 50 or so in clinical trial phase 2, phase 1 similar, and some of them have not quite reached the point where they continue on and so on. On the right, you can see these broken down by disease type. And so these I've already kind of touched on, but basically you can see the range of applications is really quite vast. So lots of promise there. So supporting vaccine quality. Okay, so back to USP's really overarching goals. So we support the development, manufacturing, and global distribution of vaccines, and we expand and make consistently available standards needed to address quality issues and build awareness on broader supply chain topics. And we do this by really developing standards, publications. We produce a number of application notes and other types of publications. We publish in trade journals, so with opinion and information coming out from USP's efforts and what we can draw from the public and so on. We expand our collaborations to provide these tools to facilitate global access to quality vaccines. And then we develop and with partners and in-house supporting analytical and regulatory capabilities for our partners. So we do a lot of this in collaboration with stakeholders to build awareness, consensus on quality. And we utilize innovative approaches to gather information and on methods and materials. So we seek interaction through a range of different interactions with the public in the forms of expert committees, expert panels, working groups, separate outreach and interaction. And we leverage the science and global reach to maximize this impact. So we also use these interactions to try to stay relevant and really at the cutting edge of where the industry is. So end-to-end vaccine quality, this is a very kind of simplified representation of the entire production process from raw materials all the way out to administration. And you can see, and I'm sure most of you are already aware, this really starts at raw materials. So quality of raw materials is important. USP targets raw materials and starting materials as possible reference standards. GMP manufacturing. So in GMP, of course, we have three major components: experience, training, and capacity. And from DS lot release all the way out to drug product lot release, we have standards that have to be in place. We have stability, data, and studies that have to be in place. Availability of quality materials to facilitate these processes and activities, and also labeling. So there's quality in labeling. And the assays developed really have to be fit for purpose. We need standards across the industry, and we need process and assay consistency. And then letting the farther after the actual drug product release, we have distribution and administration. So cold chain has to be verified, validated, storage as well. The training is also important, and administrative strategy on how we get these things accomplished. So so you can see here listed down here on the far left is kind of an areas that we really kind of focus on the raw materials, and I'll talk a little bit about that as we move forward. So moving to the messenger RNA guidelines and analytics. So the guidelines were really a new paradigm for USP. So when we were confronted with the COVID-19 pandemic, there was a need to get information out to stakeholders and drug developers and those involved in distribution and production and analytical analytics of these drugs. And so RNA is a cell-free production system, and this provides advantages as its flexibility and speed, and also generates a need for new analytical tests. So there was a novel set of raw materials and starting materials, and these were really not easily sourced and in compliance with CGMP. Novel delivery systems, unique impurities, and most PQAs, product quality attributes, will be common across messenger RNA products and therefore are amenable to platform methods and standards. So this is where we really focused a lot of our efforts. So new raw and starting materials, they would include plasmid. So we have a template, high quality DNA templates required. Enzymes or reagents, since it's sort of a enzymatic synthesis platform. We have a range of enzymes that are involved in the process, for instance, T7 polymerase, DNases, restriction enzymes, and so on. And we also have requirement for 5' capping of the messenger RNA and poly A tail. And so these were reagents that are required. And also nucleosides, native and modified nucleosides. And I'll talk a little bit about that as we move kind of high level. And we have a new delivery systems, lipid nanoparticles. So raw materials here. There's focus on size and heterogeneity, lipid content and purity, and encapsulation efficiency of these drugs at formulation. Okay, so our approach to the messenger RNA vaccine analytical guidelines. So our approach was to identify specific methods and adapt them from public sources. And again, this was to address the rapid response requirement. And it doesn't really have a direct pathway to the compendia. As you know, USP produces a range of documents, both above and below 1,000 chapters that address different pharmaceutics, including vaccines. So this really was outside that process, so we can get information out quickly. So drafted and reviewed and refined by vaccine experts. So we interacted with our USP expert committee and USP vaccine advocacy group, advisory groups, to really arrive at what became the analytical guidelines. So this is a kind of a snapshot on the left of the drop page for the analytical procedures for messenger RNA vaccine quality. You can see the link to those guidelines if you're interested in looking at these in detail. The guidelines were first shared with the public for public comment in February 2022, and they were updated last year in April at the World Vaccine Congress. And we delivered the second edition. So in route to producing that second edition, there was over 300 comments received and evaluated in consultation with our internal review and USP expert committee. And there was a range of advancements that we made going from edition 1 to edition 2. And some of those major ones included upstream methods for plasmid quality. Also, we added methods pertaining to the drug product. There was a need to identify LNP components. We provided several different methods for extraction of messenger RNA from LNP, and we added methods for other impurities that were not all present in the first edition, and also assays for nucleosides and capping reagents, and alternate methods were provided in the second edition. And then we also provided additional technical information according to the methods that were included. So at this point, this has been out for approximately 9 months. We're now incorporating another 300 comments and evaluating them and updating for a third edition. And that should be coming out soon, probably in the next, I would say, a couple of months. So this, as far as the second draft, upstream processing. So this is a list of suggested methods. We don't go into detail on these methods. Detail that would encompass the range of methods here that go across the quality and attributes. They can be found soon in chapter 1040, which will be coming out at the end of this month. And that's quality considerations for plasmid DNA as starting materials for cell and gene therapy. So that should encompass those, which would be outside of the guidelines, but would include these. So we try to include a range of methods for the range of technical capabilities over the world in laboratories that do the analytics. So a couple of kind of high-level comments about the actual drug substance and how they relate to quality attributes, identity, purity, stability, and so on. So in number one, we see here on just what is the purpose of the cap, the efficiency of the capping and the cap structure impact the innate sensing and protein production. So this has to closely match that of the host, in this case, the human host. Number two, the up and downstream UTRs. These correlate to the translational efficiency, and it's regulated by their length and their structure, and these are regulatory elements. And there's a lot of research into the different various manufacturers on producing these. The actual coding sequence. This is where we've actually imparted the code for our drug product. And then we have the poly A tail. The properties such as length are important for translation and protection of the messenger RNA from native digestive enzymes. And we're also, of course, we're concerned with purity. So removal of impurities such as residual nucleosides and byproducts and so on, and double-stranded RNA and a range of others. And so common PQAs for these products really revolve around methods that monitor the 5' cap, the poly A tail. They're essentially in a similar size range, so this is targetable as far as a reference standard or reference standards. And there tends to be aggregates, so these tend to form aggregates. So control around this is another quality that's monitored. And so, as I mentioned in a previous slide, what are some of the new common impurities? So again, residual starting materials, for instance, plasmid DNA, nucleosides, enzymes that are used in production and processing of that upstream plasmid during linearization and so on. Residual reagents and solvents, and double-stranded RNA, messenger RNA fragments, and misincorporation. So these are things we really needed to wrap around in our analytical methods, at least those represented. Okay, so this is a list of the analytical methods that are provided in the current version 2 or edition 2 of the vaccine analytical guidelines. And I won't go through all of these, but basically you can see the qualities are listed here. The attribute that's monitored is here. And then there's a range of methods, and usually more than one per attribute. So we have a range of techniques available that will encompass a range of technical abilities across laboratories. And so I won't spend a lot of time here. I think the subsequent presentation by Khalid Yamut will get a little bit further deep into some of these. These are just representatives of those analytical processes and methods. So again, we're monitoring capping, and for instance, that can be done by ion pairing our reverse phase HPLC, as represented here. Poly A tail length, which can typically be monitored by, again, IPRP HPLC with mass spectrometry. And also sizing and impurities. So, for instance, IPRP HPLC can be used here, and also, for instance, size exclusion chromatography with and without moles. And so these are kind of some of the target assays that are included, and these are actually in testing now in our labs. So these, and from those, raw materials are being processed for eventual production of reference standards, which should be compendial. So they should have eventually chapters associated. Moving on to the drug product. So these are a list of some of the common LNP quality attributes. We're interested in the particle morphology and structure, which is really related to how these things perform. Particle surface properties, mean particle size and distribution, the concentration of the particles, encapsulation loading efficiency, so what's actually inside and outside of the LNPs, the lipids, their range, their concentration, their specification, in vitro cargo release, transition temperature and stability, residual solvents and impurities, and sterility, toxicity, and efficacy. So we've targeted a range of these, not all of these. Some of them will be product specific. And again, this is a list of those that are included in the guidelines. And so again, these are the qualities monitored, the attributes, and then again, we have, in many cases, more than one test method that is described in those. Again, to encompass a range of laboratory capabilities and technical capacities. There is a list of a range of USP chapters that mostly encompass safety and other attributes. And this process requires an extraction procedure to separate the lipids from the messenger RNA drug substance, and we have provided 3 extraction methods within that chapter. I'm sorry, within that guidelines. So again, these are sort of representatives of the type of methods that are in the guidelines. So we want to look at target protein expression, Reverse phase HPLC CAD for lipid identity is one method that's in the guidelines. Also, messenger RNA size and integrity. This would represent cap gel electrophoresis, and then LNP size and polydispersity, for instance, by DLS. So next steps in our messenger RNA vaccine standards pipeline. So how are we going about this? So there's still opportunities for collaboration. We continue to encourage and engage with messenger RNA community to build a consensus on PQAs and methods. So we encourage additional comments and donations of methods and validation packages to contribute towards these efforts. We evaluate selected analytical procedures, including messenger RNA vaccine guidelines. So requesting samples for future testing to ensure method suitability. So really to test these methods, it's helpful to have samples from across manufacturers where different processes may be in place. To identify standards and controls needed to support methods. So seeking bulk materials to develop standards and so on. Develop and expand training and tools to global access to quality vaccines. So looking again for collaboration, support training, tech transfer, and receiving laboratories. So where are we as far as methods testing and material standards? So listed here are the methods that are currently in testing in our laboratories within USP. I won't go through each of these. You've kind of seen a number of these in kind of example and in the tables. And so these are 6 that are currently in process. These are really at the drug substance level. We're quickly moving to the drug product. And so then we're acquiring materials for verification and validation activities. We've now very in process now moving towards acquisition of bulks. And we're still currently assessing reference materials. So to summarize, so what can these guidelines do? Well, they can support vaccine quality by providing narrative concerning the generalized production processes and the points of control, establishing a common understanding of quality attributes for upstream intermediate, for instance, the plasmid template, the drug substance, and then downstream the drug product. And providing testing methods that can be used as a starting point to assess quality attributes, and for instance, those listed. And to provide multiple options for testing of the same attribute, again, to encompass the range of technical capabilities in laboratories that are involved in these analytics for these products. So that brings me to the end. Thank you for your attention. And I think after Khalid's presentation, I believe we'll be taking questions.
Okay, great. Thank you very much, John, for your presentation to start us off today. And a quick reminder to our audience members, if you have any questions for John, then please feel free to send in as many questions as you like via the questions tab. You can find this directly below your webinar screen. And then we'll go through these questions after our next presentation when we have our live Q&A session. Okay, so once again, thank you, John. And now please allow me to introduce our next presenter, which is Khalid Yamaut, who is Senior Director, Analytical Services and Quality Control at TriLink Biotechnologies. So Khalid is the Senior Director, Analytical Services and Quality Control at TriLink Biotechnologies, where he oversees all analytical aspects of method development and validation to product release and stability to support regulatory filings for both small and large molecules. Prior to TriLink, Khalid held various positions in quality control, research and development, and manufacturing, where he supported a number of drug substances and drug products from clinical phase to commercial. These include diverse experience and expertise ranging from discovery to manufacturing, with Fortune 500 firms, as well as small entrepreneurial businesses, in the areas of synthetic, analytical, colloidal surface modification, protein and antibody modification, and purification, covering both manufacturing and analytical testing and characterization. So with that, please let me hand over to our next presenter. Welcome, Khaled.
Thank you very much for the introduction, and thank you everyone for attending this morning. So I'm going to continue on the theme of quality assessment and what is the perspective on the future of mRNA in terms of analysis and characterization. So with this presentation, I will attempt to discuss the criticality of establishing appropriate analytical methods to characterize an mRNA, and I'll go briefly on the mRNA background and anatomy. Some of it going to probably be a little bit duplicate of what John just mentioned, so I'll probably go briefly on it. And then I will be a little bit more focused on technical stuff. So we'll look at the analytical consideration for the analysis and characterization of mRNA method, and I would like to share with everyone some thoughts about orthogonal ID methods for mRNA and poly-A, different ways to do analysis of poly-A, and also to show some interesting work that TriLink have done to establish and to make some poly-A standards that could be used for the analysis of poly-A. So the evolving mRNA therapeutic landscape, it's really taken off, and the treatments are being developed for many therapeutic activities. As you can see, there's mRNA vaccines, gene editing, cell therapy, personalized medicine, protein replacement, non-translated RNA. So the mRNA continues to be fast-growing modality, with over 45 mRNA therapies entering clinical stages. That number was from early half of 2023. Also, as the space continue to evolve, it's actually shifting a little bit beyond vaccines and COVID-19. And approximately 250 out of 800 mRNA therapies that are being investigated currently are toward the vaccine cancer indications, which is really, really good. So again, this is a different representation of the mRNA structure. You do have your 5' capping, and TriLink as part of Maravai Life Sciences. We are known for the flagship of CleanCap, which is a capping analog that gives you the Cap1. We've introduced 4 different capping agents, and each have a different characteristics that could help with your mRNA. And also then you have your 5' UTR, and then you have your coding region, the open frame, and you need your nucleosides to build that through your IVT reaction. And TriLink actually also provide all the NTPs and modified NTPs, such as N-methylpseudo-U, and all in GMP grade. Then you have your 3' untranslated region, and then the poly-A after that. So that's the basic structure of the mRNA. So now, in terms of analytical consideration for analysis and characterization of mRNA. So we here at TriLink, we do 2 things. We support our own products and also act as a CDMO. And as part of the CDMO service to support mRNA programs, we develop we follow an analytical method life cycle by phase. So we approach it via phase appropriate, and the analytical capabilities, we view the analytical capabilities to be critical to the success of all programs from discovery to market. So a well-designed roadmap ensures that the CMC data requirement is met for a successful drug development. And the model that we follow, as you see here, is, in the discovery phase, when people just trying to see if something works or not, you get your RUO, mRNA. At that time, from the analytical standpoint, we recommend to do a quick feasibility before we test and collect any data. And those are based on generic methods that we have developed for characterization of mRNA. So you're really not starting from scratch in terms of development. And then when you move on to the preclinical toxicology, it's good to ensure that you actually do the feasibility. It's not recommended. So that way, that you are meeting the intended use of the method. And this is also based on generic methods. And then when you start preparing for phase 1, that's when the methods need to be qualified. And we follow the ICH guidelines for specificity, linearity, dynamic range, accuracy, precision, LOD, and LOQ. Now, not every method requires those. It all depends on the intended use of the method. Like, for example, for an ID method, you really don't need LOD and LOQ and accuracy and linearity. And for some characterization methods, you might not need that. But if you're doing a quantitation and purity, then you need all of that. And then for late phase, that's when you add robustness, sample stability, and so forth. And during the life cycle, we actually evaluate all the methods to see if they are stability indicating methods. We look at that. And also, since we do have multiple labs, we do a cross-lab qualification, validation, and training. So that way, when the method is done, you don't have to transfer the method because the method's been qualified or validated and the training was done across all labs that are set up with this, with the equivalent equipments. So that way it cuts down on your timelines to keep your program moving. So, in terms of the attributes, I'm an organic chemist by training. Many, many years ago, I did a lot of organic synthesis, and there's something I learned there in terms of analytical that I kept carrying through proteins and now mRNA, or any molecule that is put in front of us to analyze. When you make an organic compound, you have different functional groups, and you need a different set of analytical techniques, whether it's spectroscopy or colorimetric method or something that tells you that you have the right functional group or you have the right structure. So you usually draw the structure and said, okay, if I have a carboxylic acid there, FTIR are going to tell me X, NMR will tell me Y, I can react it with something and I get this color. So you start listing those, and then you go about to analyze the structure, to characterize the structure. Well, you can do the same thing for mRNA. So this is the general structure of mRNA. And for the open reading frame, the most important thing about it is it translating the right protein for the specific pharmaceutical indication? Do you have the right sequence? And it's the functional sequence, not necessarily that, the physical sequence. So with that, you can do an in vitro translation, which is a cell-free based assay with a Western blot. If you have the antigen to bind to it, that's labeled, so you can show that you have, you're translating the right protein. Now, this is a yes or no. This is not really a quantitation and could be used as a potency assay, because for potency, you really have to have a cell-based assay. You cannot do it that way because there's a lot of stuff you need to take into account, getting the mRNA getting into the nucleus and so forth. So the next thing is, if I have the full length, what I can do with that? Well, with the full length, there's few things that might have to be done and others you don't have to do. So the thing is to keep in mind that mRNA it's way much larger than antibodies. So there's not that many analytical techniques that you can just run it straight. Sometimes you'd have to chop it into pieces and analyze those pieces, and you may have to make sure that you're chopping it correctly and all of that. But if you take the full intact mRNA, for example, you can look for integrity and purity, and you can do gel electrophoresis, capillary electrophoresis. The two main things is, the CE. The main thing is the CE. There's two main instruments that a lot of people use, which is the SCIEX and the frag analyzer from Agilent. And you can utilize HPLC, which can tell you a lot of stuff. In this case, would be an ion pair reverse phase HPLC, size exclusion, or anion exchange. Now, some people will ask, like, why so many purity methods? And I will show you later in the slides, but the reason for that is, about 40 years ago, when antibodies became used for therapeutics, the first antibody that went out is people ran a gel, saw one band and said, fantastic, it's pure. If you're in that antibody business right now and you're trying to release an antibody for commercial or, for a therapy, those antibodies have at least anywhere between 3 to 5 different purity methods, because each tell you something different about the variant of the molecule that you have than the impurity. And I truly believe that mRNA will be heading into that direction to look at different, aborted sequencing, aborted sequences, other stuff that is there, that you're going to have to characterize, but each chromatography method going to give you a different separation and different results. In terms of identity, there's sequencing, and you can, a lot of people, use Sanger or NGS. Sanger is the popular one for GMP because the software and everything is part 11. NGS require a lot of bioinformatics, and there's a lot of work right now being done to see how could that be done under a part 11 guidance, so it could be a GMP. It's doable, but it's not easy. The other two that I'm pretty excited about is fingerprinting by LC-MS and base composition assay. And later in my presentation, I will be talking about these 3 methods, as an orthogonal methods for ID. For content, you can use UV spectroscopy or IPRP, HPLC if you do a standard curve with your mRNA. For capping efficiency, we developed an LC and LC-UV method, and the same thing for the poly-A tail. And for impurities, DNA by qPCR, residual DNA by qPCR, protein, residual protein by nano-orange or BCA, residual NTPs by anion exchange HPLC method that we've developed. If you use any solvent, GC, USP method, 467 would work, and double-stranded RNA with immunoblot or ELISA. And, your safety method, endotoxin and bioburden, and some people request sterility depending on how you're using your mRNA as a final drug product or as a DS. And others, appearance pH, but you can put conductivity and all other methods to help you with downstream process formulation for your DP. So gonna move on now to actual methods and data. So this is a quick case study that we've done here at TriLink to show an orthogonal mRNA ID methods and what each ID method can tell you about your ID, and if you use them in combination, what you're gonna get. So the 3 methods that we looked at is the sequencing by Sanger, fingerprinting by LC-MS, and base composition by LC. So the case study, what we did is we used an F-Loop mRNA that one was made with a wild type NTPs, and one we actually mixed some uridine and N-methyl pseudouridine and made the mRNA as a mixture of uridine and N-methyl pseudouridine. So the idea here is if you have a modified mRNA, or you have modified bases in your mRNA or a completely modified mRNA, does the ID method gonna tell you just the sequence, or would it tell you also what the composition is? Do you have the uridine or the N-methyl pseudouridine in this case? So So for Sanger, we designed the four-way primers and followed the normal procedures that a lot of people use. And for the control, which is the fluke wild type, we didn't see any mutation detection, and the sequence was a match. And for the fluke that was made from the uridine and methyl pseudouridine mixture, we saw the same thing: no mutation detected, and the sequence was a match. And here I tried to show a representation of the sequence coverage, and you can see it cannot tell you which is pseudouridine or which is just uridine, or a mixture of uridine, pseudouridine, and a control. So Sanger is really good to tell you exactly what's your sequence in the ORF, and do you have the ACGs, but it doesn't really distinguish between an NTP or a modified NTP. So we also have developed a fingerprinting LC-MS method. And the way this method works is it's a little bit based on some of the stuff that was published by Thermo Fisher, where we did a partial mRNA digestion using magnetic beads with immobilized RNase T1. It's a controlled digestion because you don't want a full digestion. If you have a full digestion, you end up with a lot of fragments, and analyzing the data would be close to a nightmare, and won't tell you that much. We incubate the reaction between 2 to 30 minutes, depending on the construct. Then you separate the mRNA fragments from the magnetic beads, analyze by LC-MS, and then we analyze the data using the biopharma finder software. And in parallel, you have the in silico to the experiment, where you put your RNA sequence, in silico digestion, what would be the digestion product and the theoretical precursor, and then you match the two to give you scoring and ranking, and map the modification into mRNA sequence. So this is an example of what you get in terms of raw data. So we applied that on 3 different mRNAs just to look at the length, the impact on the length on the method. So we used EGFP, which is about 996 bases, fluke 1929, and Cas9 4521. And we got very, very good coverage. And the coverage that is the sequence coverage that you're looking at is the ORF plus the UTRs. It does not account for the poly A. So you can see the coverage here is 90 for EGFP, 97.9, and so forth. But if you account for the poly A and take those, that out, it's almost 100% coverage of the ORF plus the UTRs. And we showed that this data is reproducible by running 3 lots. And you can see the nth, for the 3 lots, the RST is below 1.1%, which is actually pretty good. So now when we looked at that data, it's like, how could you present that data to show that you have the right ID? This is great, but this is a lot of little details that you have to dive into to extract it, and you're gonna find a way to present it. So one of the things that we came up with is to present the data as a correlation curve. So we assigned one of the 3 lots to be our standard, or our, that's the material that we tested against. And we plotted the other 2 lots against that. So as you can see, for the Cas9, the EGFP, and the fluke, for all 3 of them, you get an R-square, the 1. It's a perfect match. So that tells you that the ORF, the sequence of the coverage sequence of the ORF with the plus the UTRs matches what we're calling a reference. And if we take the data and plot the Cas9 and the fluke, the EGFP and the fluke versus the Cas9, you don't get that very good correlation. So that tells you that your mRNA is not exactly what you're looking for against your standard. So this was a good, a good way to see the data and present the data for that way this method could be used as a release method with an easy with a thought, with a way to present the data in a I don't want to say in an easy way, but it's more representative way that you can score yes or no. So when we applied this to the uridine mixture, uridine and modified uridine mixture versus the wild type flu control, as you can see, we get a good correlation on the top graph. But then we said, let's go back and look at the tick, and because the previous slides, this here, for this slide, what we're actually showing is the tick chromatogram. And the tick chromatogram didn't show much. So we said, how about we go back and look at the UV chromatogram? Because, when we run this, you run a UV and then mass spec. And we saw some slight differences between the control and the uridine with the mixture of the N-methyl pseudoU. So you can see in black here, this is the sample that contained the uridine and the N-methyl pseudouridine, and then the control. And you can see that little peak right here that correspond to uridine, and the high peak correspond to the N-methyl pseudouridine. Now, if you did not know that N-methyl pseudouridine, it's kind of a little bit difficult to tell, but it tells you, if you look at that in addition, it tells you that the two mRNAs are not the same. There's something different about them. Even though the sequence coverage might be the same, there is something in the composition about them is not the same. So then we went old school. And when I talked to someone about this method, they said, oh, you're going really, really old school on us. So what we have done is, this is a method that was developed to check the base composition of the mRNA. So we took the mRNA and digested to the phosphodiesterase, and then we converted the nucleosides. We converted to nucleosides by treatment with the CIP, and then we analyzed and quantitated the sample for the nucleoside bases, the basic base, by reverse phase HPLC against a standard curve. So here you can, on the right-hand side, you can see this is generated calibration curves for the C, U, G, and A, and we have a linearity range between 0.4 and 200 micromolar. So here's a chromatogram of the base of the assay, where the black is and this is slightly offset. So this is why you see in two peaks. If I don't offset it, they kind of land on top of each other and you won't see anything. So the wild type mRNA control is in blue. So you can see your uridine, the C, the U, the A, and the black is the mRNA that made with a mixture of N-methyl pseudo-U and uridine in black. And we can see the N-methyl pseudo-uridine peak, the uridine. So there's a nice separation, so you can actually quantitate it. And if you zoom, it's really a baseline separation that you can actually quantitate how much of each you have. So here's just the curves that were generated and the ranges of the concentrations. And here another set of curves, but we added the N-methyl pseudouridine so we can quantitate. So from all this is the final chromatogram that was generated, and this is how much micromolar there's present of each base. So now if you do the calculations, the Fluke wild type theoretical, what we're supposed to get is, this is how many C's used, G's, and A's. And these are the two mRNAs that we've made. We made them together at the same time. So you can see the C is pretty much identical for the two. That means the process at the time, the IVT, it's pretty reproducible. It's slightly off from the theoretical. It's less than 5%, but that is within the error of the assay of the, in terms of, we have not done a lot regarding the quite a bit of quantitation against the theoretical to see what was the differences, but that's within the assay error, I would say. And you can see for the control, we see the wild type the U, and for the mixture, you can see there's a 26 micromolar of the uridine and 242 micromolar of the N-methyl pseudouridine. And if you add those up, they'll equate to 264, which is pretty close to the 262. And you can see the G's are about the same and the A is about the same to give us a total of 1922 for the total length. So this method is not going to tell you the sequence, the order of sequence, but it'll tell you, it's very accurate in telling you what composition you have, just in case you see something different with your ID. So as a summary, for orthogonal mRNA ID methods, each ID method provides a unique information regarding the identity. So as I mentioned, the Sanger sequencing provides a sequence coverage of the ORF that would not directly differentiate between wild type and modified nucleotides. The fingerprinting LCMS, give you the sequence coverage of the ORF and the UTR, and indication of any possible nucleoside mismatching. And the base composition provides specific detail regarding the composition of the sequence and how it is matched to the theoretical sequence-based composition, but it would not tell you the sequence coverage and the order. So if you look at it, if you combine the three, they can provide a very powerful tool for investigation and some detailed level of the ID testing. And I can envision futuristic that the base composition and one of the sequencing method would be your two ID methods to release a commercial product in the future. Next, I'm going to jump a little bit into purity by ion pair reverse phase HPLC. This is a method that we developed a while ago for purity. And here what it's shown is five different mRNAs that we ran and overlaid. The difference between this and CE is the CE was separated by size, but this is really not separating by size. There is some interaction between the column and the molecule, and that really impacts the retention time, and hence it gives you different results from CE. And here, those mRNAs were picked up on purpose just to show some of the impurities. So that way, as we develop further the method, we were able to look at impurities and how we can analyze those impurities. So again, on the theme of orthogonal methods, between HPLC and CE, which one you want to choose? So what we've done is we made our own standard mixtures of EGFP, FLuc, and Cas9. And we analyze it by frag analyzer and IPRP HPLC. So to your left, you see the frag analyzer, and to your right, you see the HPLC. And when someone look at this, the first question they'll say, like, why do I want to use an HPLC? I don't have a baseline separation. Those are really beautiful peaks. HPLC is a waste of my time. Well, my question going to be is this the peaks in the CE, the non-sharp peaks, are they really one peak, or there's something hidden under there that you're not seeing? Or can it tell you, or can the HPLC tell you something different? So one of the things is that you encounter is aborted sequences, and as part of it, you can have some untailed mRNA. So we analyzed only one species of untailed mRNA, which is the full-length untailed mRNA. So we took an FLuc mRNA that we've made. We made the full length with the poly A, 120A, and the one without the tail. And as you can see by IPRP HPLC, we can see a baseline separation between the two. You look at the right-hand side, and we were down to 0.5% untailed spike, and it still shows far, nicely separated from the full length tail. Well, at 90%, 10% spiked into the tailed, which is 90% tailed, you still see only one peak. we have to spike in 30% of the untailed to actually see a shoulder peak. So, this is expected with CE because you're just changing the size, the molecular weight by 120 A's. That might not be enough to actually see two peaks. So this becomes very hard to quantitate purity by CE, and in this case a frag analyzer, because you're not exposing all your aborted sequences and all of that. So you might see a lower purity by HPLC, but it'll be a much more accurate purity, and that'll help you to get a much purer mRNA. The same thing if you have two different tails. For some reason, you got two populations of tails. In this case, we've made a Cas9 wild-type mRNA that have an 80 and 120. And you can see you can separate the two by HPLC. You get a different retention time. No matter how we mix them with the CE, you still see only one peak. And again, that's a difference of 40 A's. That's not enough molecular weight to actually see two peaks and separate it. Same thing with base modification. Base modification, in this case, we made mRNA with the 5-methoxy-U and wild type, and you can see clearly there's a separation between the two different mRNAs with the different bases, and you really cannot see it with the frag analyzer. It does not separate them. So with that, we took that method also, and we showed the capability of being quantitative by getting a standard curve. So you have an IPRP method that is specific. You have a nice linearity, and the precision is below 2%. So next, I'm going to jump into poly-A. So due to the size of mRNA, direct analysis of mRNA by liquid chromatography mass spec can be challenging. So as such, it requires to digest the mRNA prior to analysis. So the main methods for digestion is, one is an annealing methodology using DNAzyme. And in this case, you anneal the DNAzyme at the 3' UTR, which will yield the poly-A plus X bases and the untailed or the cleaved mRNA minus X bases. And the X bases all depend on the design of the DNAzyme that we've made to anneal to the 3' UTR. The other way you can do it is you can do enzymatic digestion, such as RNase, which will yield a complete digesting of the mRNA except the poly-A portion, and then you can analyze for the poly-A. Or we actually were able to do some work with the base composition to tell us a little bit about the poly-A. So once the samples are prepped, then they can be analyzed by a wide variety of analytical methods. So also recently, various publications on the subject and quite a few approaches are still being vetted and evaluated for fit for use in the QC environment. So again, this is a schematic of the mRNA. This is showing the RNAs A and T1 as a combination, or each digestion. You get all the mRNA, the fragments plus the intact A, and you analyze it directly by LC-MS. This is what you're getting here is a representation of a chromatogram of the sample, where you get the digested fragments and the poly-A. You deconvolute the data, and that'll give you a poly-A distribution. In this case, we're targeting 120. Your distribution here with the average of 124. That's within the acceptable range of what the poly-A should be. And again, this is not a quantitative. This is more of a characterization assay to tell you have the tail and what is the distribution size of the tail. So one of the questions we ask is, can this differentiate between two poly-A tails? So if I have two populations of poly-A in my sample with this method detected. So that's kind of similar to what we've done with the IPRP. It can separate it, but here, again, we took a mixture of the Cas9 80 and 120. And you can see by UV, you can see a nice separation between the two. And the tick chromatogram, you had two peaks. You deconvolute the two peaks. The 13.5 is your 80, and the 13.8 is your 120, which is in this case 124, and this is supposed to be 80. So again, this is within the acceptable range of your poly A distribution and size. And lastly, we worked on determining the poly A tail by base composition. The other method was base composition. So as since we developed this ID composition assay, so we said, can we determine poly A with that? So we took six lots that we've made of fluke mRNA made by the same IDVT process and the backbone, and we analyzed them against an mRNA without a poly A. So it's just the ORF, the 3' UTR, the 5' UTR, and the capping with no A's. So the sample were prepared and analyzed, and the poly A length was determined based on the concentration of the A's, and that was compared to the value of the A by LCMS method. So the observed A that we saw for the fluke mRNA, for all the lots is 48. This is what we got. And the theoretical A's for this fluke without the tail would be about 380. So from that, the average tail using the composition method, they're going 118, 116, 118, 120, 122, 124. So it's within the range. How did this compare to the LCMS method? So we took the same 6 mRNAs and we digested them with the RNAs and T1, and we analyze it, and by LCMS we get an average of 124. So this also can give you an idea about your poly A, but also shows a little bit about the dynamic range or the accuracy of the percent error of the composition assay for that. It's slightly larger, but it's still giving us the same, it's within the range that you would expect for the A's to be there. The last is the direct analysis of the poly A through DNA enzyme cleavage. So what we have done is in this case, we took an EGFP and we designed a DNA enzyme to anneal at the 3', and we annealed it and cleaved it, and then injected it in, and did the LCMS analysis. And this here shows the UV scan of a IPRP method, so where we get a full-length untailed poly A in the DNA enzyme. This IPRP method is not mass spec friendly, so we developed another method that is more mass spec friendly. In this case, this is a reverse phase HPLC method, and you can see here the cleaved mRNA and then the tail. Now, the difference between the two methods is this IPRP will actually separate the tailed and untailed. The LCMS method that we've developed, it cannot separate the tail, the cleaved or untailed from the full length intact mRNA, but it separates nicely the poly A for analysis by mass spec. So in this case, so this is the thick mass spec, and this is the cleaved, and this is your poly A. It's around 127. So is this really what we're looking for? So we did a sanity check, and we've asked our oligo team. As you know, TriLink started as an oligo company before they moved into mRNA and capping and so forth. So they made us a synthetic 120 mer poly A, and you can see this is the tick chromatogram, and if you compare the retention time, we're pretty. It's a good sanity check that we got the right size. Now, some people will say, well, this is 127, this is not 120. Yes, and that is expected because you have to account for the bases that were used to anneal the DNA enzyme to cleave it. So remember, you're going to get the poly A tail plus X bases depending on the design of the DNA enzyme. So this is the deconvoluted. So this is the deconvoluted spectra of the tick for the 120, and you can see it's a really nice one sharp peak. And this is the deconvoluted with the distribution. It's giving us around 130. So with that, I showed multiple ways to analyze the poly A, and they are orthogonal to each other. So if you combine all the data, you can see each method gave you something that you can compare and tells you something something about your poly-A. So finally, I want to go really quickly through this synthetic poly-A standards that we've made. This was a internal cross-functional collaboration between multiple teams at TriLink to make those. So we made 5 synthetic poly-As, 39, 59, 79, 99, and 120. And we analyzed them by 6 different chromatography methods. So here is an overlay of each standard versus a mix, with against the mixture. The mixture is in orange of the standard, and we can see separations. And this is just an internal HPLC methods that we have. It's an HPLC-LC method actually, and then we looked at the tick chromatogram and we deconvoluted, and the LCMS area percent, the purity is above 95% pure, or for all of them. The nice thing is, if you look at the chromatography, this is the poly-A mixture standard curve that we generated to see what would be the perfect fit for this method. And we have the correlation is, you get 9.6 when you use a log versus retention time, the log length versus retention time. What you see here is, what I brought attention is the nice sharp peak. There's stuff in the literature that you do not see this nice sharp peak without a major tailing on poly-A standards. And another publication that was out late last year where they claimed they could not make the poly-A standards. Instead, they made a pseudo poly-A standard by a sequence of 19 A's, and they inserted a G after every 19A. So people need to be careful using those kind of standards, because you might not be able to account for secondary structures or anything like that, because that would not be a true representation of a true poly-A full length. So as a result, these are the standards that we have: the 120, 99, 97, 59, and 39. All of them are, the percent area of the method is pretty high. They're very high purity. Now, we took those and did a standard curve on using 5 different LC methods. So we did the octylamine method, which is based on the Waters octylamine column instead of using IPRP. We used AX method, IPRP, HPLC method, size exclusion, and we do have a poly-A generic LCMS method that we did that. So all of them, in a way, we were able to generate some kind of standard curve to identify the length of the poly-A. I would not say quantify, I would say identify the length. So what we have done is we took 2 mRNAs, one Cas9 with 80, and the second one is EGFP with 120. And we analyzed the poly-A by the 2 methods that I've mentioned above. One is by the RNase digestion and one by the DNA enzyme cleaved. So that way we can calculate the theoretical of those two mRNAs before we analyze them with different methods using the poly-A standards. So the Cas9 digested gave us an 80 and the cleaved gave us 5, which is accurate because there are 5 mer of the 3' UTR region with the tail. So they are equivalent. And again, the same thing with the 120. We had to use a different DNA enzyme for this one. It's a 7 mer, and you had 120 and 127. So now we have the theoretical value to compare from an HPLC method. So now we analyzed both of them, the digested and the cleaved by the octylamine, anion exchange, SEE, and mRNA and the generic poly-A. And you can see, for example, for EGFP with the SEC and the generic poly-AMS, you get the closest number for theoretical. For the Cas9 digested, again, the SEC, you get a mRNA with the IPRP, the closest. The EGFP cleaved, you're getting the octylamine or the AX to be the closest, and the Cas9 cleaved is your AX is the closest. Now, those were one runs. We haven't shown a lot of reproducibility and so forth, but that gives you an idea that now that opened the door to a huge amount of tools and possibilities of how to analyze different things and how to use these standards. And also you have to be careful on the method that you're using before you start reporting your tail, because you really need to characterize it properly. And this is where orthogonal methods become really important. So with that, I would like to summarize. We have robust analytical methods for mRNA characterization. I've showed orthogonal ID methods and multiple approaches for poly-A analysis and the standards. Here, at TriLink Analytical Service, we provide a full service for mRNA characterization, including identity quantification, product characterization, functionality, impurity profiling, and we can achieve your analytical objectives from process development to scale up to GMP manufacturing. And this is the expansion of the analytical service for the quality and the quality control lab that we did last year. And recently, last September, we actually launched the Analytical Sciences Center of Excellence. So this will be a centralized hub to derive innovation in nucleic acid analytical methodology. We developing additional methodologies for characterization of nucleic acid. We provide method development, qualification, validation, stability testing, and product characterization. And we also offer standalone support for RUO and non-clinical mRNA for a lot of companies that they experimenting with making mRNA, but they don't have the analytical tool to characterize them. So we offer them that to help them out with their programs. And none of this work could be done without a great team. And this is this picture needs to be updated, but we really have a really good dedicated team to generate all this data and work on all these methods. And with that, I'd like to thank you and open it to any questions.
Okay, great. Thank you very much for that, Khaled. Great presentation. And a quick reminder to our audience members, if you have any questions for Khaled or for John, then please do send them in via the question